Welcome to Virtual Cell Tutorials. Go to vcell.org to download the software. In this tutorial, we create a single biomodel of RAN nuclear transport events and then use different applications to solve simulations with different strategies. This multi-app tutorial is presented in two parts. In part one, we develop the model and then a spatial deterministic application where we use a three-dimensional geometry to create a deterministic spatial simulation. In part two, we develop additional applications, including compartmental applications that include parameter estimation and stochastic spatial applications. So in this first part of the tutorial, we will first create a biomodel physiology. Then we will create a spatial deterministic application of the physiology. For the geometry, we will import a fluorescence image stack into VCell and segment that stack to create the geometry. Finally, we will create a simulation, run the simulation, and view the results. If this is your first time using VCell, use the new user registration to register as a new user. This will allow you to run simulations on VCell compute resources or use the VCell database to store your model and share your model with collaborators or with the public. Select the new user registration box and provide a username, password, and email address. VCell opens with a blank biomodel interface. If you want to create a new model, go to File, New, Biomodel. To create the biomodel physiology, we first need to create the compartments. Click on the Compartment tool in the main workspace window. Two dashed lines will appear at the edge of the compartment. Right-click on one of the dashed lines and it will turn green. A menu will appear. Choose Add Membrane. To add a volume compartment, right-click on the dashed lines and select Add Compartment. To rearrange the order of membranes and compartments in your diagram, select the label of the compartment or membrane and drag using the mouse. You want to continue creating compartments and membranes until you have those necessary for the model. Extracellular, cytosol, and nuclear volume compartments separated by plasma, PM, and nuclear NM membranes. To rename compartments and membranes, use the Selection tool to double-click on the name in the diagram, or type the name in the box labeled Structure Name in the Object Properties panel at the bottom of the window. Type any annotation you wish to include in the model in the annotation box. Now that we've added all the compartments and membranes, we can begin adding the reactions and fluxes. For our model, let's first create the flux across the nuclear membrane. Select the Flux tool from the tool menu and click on the nuclear membrane where we want the flux to occur. Now select the Reaction tool, click in the cytosol, and drag a line to the flux icon. A reactant species is created in the cytosol. To create the product, click on the Flux tool, then drag a line to the nucleus. A product species will be created. Now let's create a reaction in the cytosol. So click on the Reaction tool again, and then click on the species in the cytosol. Then drag a line out anywhere into the compartment. A new reaction node will be created. Create the product by again clicking on the reaction node and dragging out into the cytosol. To rearrange the positions of species and reactions, use the Selector tool to select the object and drag it around within the compartment. Select the object and use the Delete button to delete an object. Continue creating reactions and species until you've completed your model. To rename a species, select the species, then type in a new case-sensitive name in the Object Properties box. Now we need to specify a reaction rate for our flux reaction. Select the flux reaction and go to the Object Properties panel, and we need to type in an expression for the reaction rate description for J. We type Kflux times the difference between RAN-C site and RAN-C in the nucleus. This makes the flux rate dependent on the difference between RAN in the nucleus and RAN in the cytosol. The parameter Kflux that we defined in our flux reaction will appear in the table as a new parameter. To edit that parameter, Simply double-click in the expression box and add in the value for that parameter, in this case the number 2. For the reaction in the cytosol, we can keep the default mass action kinetics described in the expression box, but we do need to add in the parameters for the forward and reverse rate constants. Again, double-click in the expression box in the row for the forward rate and add the appropriate expression. Then type in the appropriate expression here, 1000, for the reverse rate constant. The physiology of your model is now complete. You can use the Reactions, Structures, and Species tabs to look up specific details of the physiology. This is often useful when working with large and complicated models. Now we need to create an application of the physiology. Use the Add New button and select Deterministic. In this tutorial, we will be creating a spatial application, so you will need to download an image set for your geometry. Go to our website at vcell.org and then click on the support page. The images are found in the tutorial guides table. You can click on the multiple application of nuclear transport part 1 3D images. You can download the images and extract the files and save them to your computer. Our first application is a spatial deterministic application, so we need to create a geometry. Click on the Geometry tab and the Geometry definition, and then click on the Add Geometry button. Then select New. From the dialog box that appears, select Image-Based and then OK. Navigate to the folder that contains the images you downloaded. Click on the folder containing the images and then select Open. You have the option of reducing the file size by reducing the resolution of the images. Holding down your cursor, adjust the slider to the desired scale for the factor for the images and then click OK. The images that you downloaded are directly obtained from the microscope and are not pre-segmented. So we will need to segment and define which pixels belong to which domains in the image. So when the image first opens in the VCell geometry viewer, we select Add Empty Domain to begin adding pixels to a particular domain of the resulting geometry. We will begin assigning pixels to the nuclear domain. So in the Enter New Domain Name, we can enter Nuke, then OK. While you're working on the geometry, you can view different focal planes by using the slider to step through different slices of the image. To change which domain you're viewing, pixels assigned to, select a different active domain. To reduce the size of the ultimate geometry, you can reduce the size of the image to contain just that of the cell of interest. Use the mouse to select the crop tool from the right toolbar beside the image, and then drag the mouse to outline just the areas of the cell that you want to include in the geometry. An automatic crop tool is also provided that automatically crops only to non-zero pixel values. Now we need to segment the image, or that is, to define which pixels belong to different components of the eventual geometry. So to do that, we'll use the tools to help us identify the appropriate pixels. To begin with, use the magnifying tool to make the image of the cell bigger. Apply a smoothing filter to the image using the scroll bar just above the image. The histogram tool below the image provides a way to select pixels based on their intensity values. Click the mouse and slide it along the values that you choose to include within the domain. All pixel values selected in the histogram tool will be identified and selected in the image. With a 3D image, remember to use the Z slider to move up and down in Z to determine whether the correct pixels are being selected. If you're happy with the pixels that are being selected, then select Apply. Note that at this point the selection does not need to be perfect. There are some pixels within the nucleus not selected and pixels outside the nucleus that are selected. These will be edited in the next couple of slides. In the dialog box, select Update Domain to update the currently active domain, which is the nuclear domain. To see all the adjacent pixels that are highlighted but should not be part of the nuclear domain, click on the largest domain of all the regions located to the left of the image. Everything in that region will be turned orange. If there are things outside the nucleus that are orange, then you need erasing. Use the eraser tool to erase regions of the image highlighted that are not to be included in the nuclear domain. As you drag the eraser tool over the image, those regions erased will no longer be highlighted. You can change the size of the eraser by either increasing or decreasing the magnification of the image, or right-click and select Brush Size Manual. Scroll through Z and continue using the eraser to erase regions of the highlight that are close to or touching the actual nuclear region that you want as part of the domain. To remove all the non-adjacent highlighted pixels that should not be part of the nuclear domain, use Control-A to select all of the domains within the domain region list, and then holding down Control, click on Nucleus and Background to deselect those two. Then select Auto Merge. This will merge all of the selected domains with the appropriate adjacent domain. In this case, it will be the background. Now that we've segmented the nuclear domain, we can move on to segment the cytoplasmic domain. Select Add Domain and enter the name Site for the cytoplasmic domain, then OK. With the active domain depicted as Site, once again we can use the histogram tool, sliding the cursor along those pixel values that we would like included within the cytosolic domain. Once again, make sure to go through all the Z sections in order to make sure that you have the correct pixels highlighted. Once you're satisfied that you've picked the appropriate pixels to form the new domain, then select Apply and say Update Domain. A dialog box will appear with a warning that some areas of the new domain overlap with existing domains, that is the nuclear domain we defined earlier. Select the Keep Existing Domain Regions When Overlapping button to maintain the nuclear domain and simply assign to the cytosol pixels that are not in the nuclear domain already. To remove pixels that were highlighted but do not appropriately belong in the cytosolic domain, hold down the Control and A key and select all of the domain regions to the left of the image. While still holding down the Control key, click on the largest regions labeled Background, Cytosol, and Nucleus to deselect those regions. Then use the Auto Merge function again to merge the isolated pixels with all of the neighboring pixel domain assignment. This will leave you with 3 defined domains: background shown in black, cytosol highlighted in green, and nucleus highlighted in red. Again, scroll through Z to make sure that the assignments are all appropriate to the image. If it all looks correct, then select Finish. A dialog box will appear asking what to do with pixels who have not been assigned to a domain. In this case, we will choose Assign as Default Background. In our example, another dialog box appears warning that some of the domain regions touch the boundary of the geometry. This is important to consider, as if a compartment touches the domain boundary, then it becomes open to the infinite space beyond the domain boundary and will affect how the simulation occurs. If you want the compartments to be fully contained within the geometry, then we need to assign some border pixels to provide a space for the boundary between the two regions. So select Add Empty Border. This will add extra pixels on the X and Y boundaries, as well as adding an additional entire plane of pixels at the top and bottom. Now that we have a geometry, we can visualize it with our geometry viewer. To see the geometry in 3 dimensions, click on the Surface View tab. Here you'll notice that it's a very flat object, and that is because we haven't appropriately defined the geometry sizes in X, Y, and Z. So select Edit Domain and type in the correct sizes for the geometry in the dialog box that appears. In this case, the geometry is 100 micrometers in X and Y and 26 micrometers in Z. You can also adjust the opacity of the different regions so that you can see underlying regions better. For example, in this case, so you can see the nucleus better, use your cursor to adjust the slider to the left of the image. Your geometry is now defined. Now we need to map the regions in the geometry to structures within the model. In the geometry, select the Structure Mapping tab and then select the Line tool. Click first on the structure in the physiology diagram and drag the line to the appropriate color for the region you wish that structure to be associated with. Do that for each of the volume and membrane domains in the model. Once the geometry mapping is complete, we can move on to identifying the initial conditions for this simulation. Click on the Specifications tab, and we can begin by providing initial concentration conditions for each of the species. Double-click in the Initial Conditions box and add the appropriate values. We will leave the diffusion coefficients for the species at the default value of 10. Now we're ready to create a new simulation. Select the Simulations tab and then the New Simulations icon at the top of the window. Select the Edit Simulation icon. You can view the parameters for the simulation under the Parameters tab, but we'll move first to the Mesh tab to define the resolution of the geometry for the simulation. To maintain the aspect ratio of the simulation, we want to ensure that the Lock Aspect Ratio button is checked. Then we can edit the mesh size to ensure that the X and Y values are at 101 and the Z value is at 27. Then select OK. Now select the Solver tab and add in the appropriate values for starting and ending time, the maximum time step, and the output interval. That is how often you want to save the simulation results within the database. In this example, we have a maximum time step for the calculations of 0.1 second, but the output interval we only save every 0.5 seconds worth of data. This reduces the amount of time spent in saving data and the amount of storage needed to save the simulation results as a whole. When you're done, click OK. A dialog box will appear with a warning of the large size of the simulation. In this case, just say OK. To run the simulation such that it will be saved automatically to the VCell database, click the green Play icon. The blue icon will also run a simulation, but the data will not be saved to the database. The status of the simulation run will appear under Running Status. As soon as there are data saved to the database, you can begin to view the simulation results even before the simulation has been completed. Click on the Results icon to view the simulation results. The results viewer shows an XY plane of the image geometry for a particular variable at a particular time. To change the Z position of the stack, move the slider in the box at the lower right of the window. Change the time using the slider in the upper left, or type the value directly into the uppermost box. You can toggle between a color or grayscale lookup table in the data range area. A number of tools are available to plot or visualize the data. To create a time plot, select the Dot tool and then click on a number of different points within the simulation. From the Plot drop-down menu, select Time. A plot of concentration versus time opens for each of the positions that you selected. To change which species are being viewed, simply select them from the Y-axis list. That completes the deterministic spatial simulation. In Part 2, we develop compartmental applications, including parameter estimation, and also some stochastic applications.